I am going to talk about bacterial senescence. Do bacteria get old? That is the question. In eukaryotes, especially mammals, aging is easy to observe. This young mouse eventually gets old, and that makes it more and more likely to die of something. The causes of aging are still debated, but one of them is thought to be telomere shortening. Every DNA duplication makes chromosomes end shorter. Eventually, a cell becomes unable to divide itself anymore, and that cell is considered senescent. This limited number of cell divisions also happens in yeasts. For example, in Saccharomyces cerevisiae, mother cells stop budding after 10 to 30 replications, and they even start to look old and wrinkly. But what about bacteria? Could senescence even happen? Prokaryote chromosomes are circular, so there's no telomere to shorten. If telomere shortening is the cause of cellular senescence, it can't happen in prokaryotes. And there's more. Many bacteria reproduce by binary fission, which looks symmetric. If these two daughter cells are identical copies of the mother cell, and they both get older over time and eventually senesce, that would mean the entire population of this strain is getting old. And that most likely means a quick extinction. Since that is not what we are seeing, maybe senescence doesn't happen in bacteria. And for a long time, experts assumed that too. But in the last 2 decades or so, things have changed. In 2003, decreased fitness over time was observed in the budding bacteria, Colobacter crescentus. In this species, mother cells are called stalked cells and stick to a surface, and daughter cells are called stroma cells and move freely with flagella. Since these two kinds of cells are morphologically distinct, it's easy to keep track of the mother's age. Later in 2005, a similar decline was reported for Escherichia coli, which reproduces by binary fission. The authors paid attention to which side of the cells the new cell walls were formed, and called that side of the cell its new pole, and the other side the old pole. If the daughter cell that emerged from the new pole of the mother cell is considered to be a newborn, we can keep track of the mother cell and its age and number of replications through cell divisions. The idea here is that binary fission only looks symmetrical, but it's actually the same process as budding, when one is the mother cell and the other is the daughter cell. The two cells that emerge from a cell division are not like twin daughters as people used to think, and the mother is older than the daughter. In other words, binary fission is morphologically symmetric, but physiologically asymmetric. Observations under this assumption revealed that the older E. coli cells had a reduced growth rate, decreased rate of offspring formation, and increased risk of dying. It sounds like they age, but this decline in growth and reproduction was not replicated in some later experiments. So some are saying this might be some kind of stress response. The exact mechanism of senescence in bacteria, or any organism for that matter, is still unknown. But most current research seems to be focusing on the loss of proteostasis and epigenetic alterations, among many other possibilities. Whatever the case may be, I think discoveries in this field will have interesting applications because if the mechanism is in common with metazoans, we can use that knowledge to treat many of the deadly human diseases, and if not, maybe we can take advantage of that mechanism to develop new kinds of antibiotics. Or something like that. You can find the references in the description. Thank you for watching.